Thanks for the introduction. So good afternoon, everyone. The research I'm presenting today is a study we've recently conducted where we examine the question of whether we can potentially use a cognitive task to individualize the site of stimulation at the left dorsolateral prefrontal cortex for using TMS to enhance cognition. So as you're aware, cognitive impairment is a common consequence of many neurological and neuropsychiatric conditions for which there's very limited effective treatment options available. And so in this context, as we've heard about in the sessions today, there's been a lot of interest in the use of TMS as a novel intervention for cognitive enhancement. And so the idea behind this study is whether we can develop a new methodology to enhance the efficacy of TMS for cognitive enhancement. So ever since the first controlled clinical trials were conducted of TMS in the late 1990s, there's been interest in whether a therapeutic course of TMS can produce cognitive enhancing effects. Our team investigated this question in more detail in a systematic review and meta-analysis that we published a few years ago, where we examined this question in patients with depression. As you can see, what we found was for two of the outcomes that we examined, the trail-making test part A and the trail-making test part B, we found a significant effect of active TMS compared to sham for improving performance. And so these tests assess visual scanning, processing speed, and cognitive flexibility. As you can see, though, the effect sizes were quite small, which raises the question of what can we do to enhance the efficacy of TMS for cognitive enhancement? So one approach that's gained a lot of research attention recently, as we heard about in the symposium earlier, is improved targeting. And this has been found specifically to be particularly relevant for treating depression. So I have here a figure that I've borrowed from Robin that he presented earlier. So this is from a review article published last year, which shows the evolution of TMS targeting for treatment of depression. So you can see that where we started off using standardized scalp measurements, we then moved on to structural scans to individualize the site. And now more recently, we're looking at functional connectivity and functional neuroimaging to individualize the site of stimulation for treating of depression. So kind of like the rationale behind this study is whether we can use an alternative functionally based approach without neuroimaging, but just using a cognitive task to individualize the site of stimulation for using TMS for enhancing cognition. So the question we then faced was, what task should we use to do this? In our search, we came across a series of studies that were conducted in the late 1990s, which investigated the effects of TMS on the random letter generation task. And briefly, what this task involves is approximately every 1 second, people have to say a letter of the alphabet between A and I. And what this task assesses is cognitive flexibility. So firstly, what this group showed was in a functional neuroimaging experiment using PET, was that the left dorsolateral prefrontal cortex was significantly activated during task performance. They then showed in a series of studies that when you apply active online high frequency TMS during task performance, where TMS is applied immediately before people provide a response, that this reduced people's performance and made people perform less random. And this was specific when stimulation was given to the left dorsolateral prefrontal cortex. So in our study, we investigated this further in a crossover sham-controlled randomized experimental design. It's a single session study in 25 healthy participants. So we adopted the same or very similar experimental methodology that was used in the early studies in the 1990s. And that's what's shown here. So firstly, participants performed 2 blocks of this task with no stimulation. Then they received active or sham stimulation online high frequency TMS administered to 5 standardized sites at and around the DLPFC centered around F3. During each trial, participants received active or sham high frequency TMS given at 20 Hertz, so 4 pulses, which were administered immediately before every second required verbal response. So the two outcomes we're interested in are called unique diagrams and trigrams. And what these represent is just unique combinations of letters. So either 2 letters or 3 letters, which indicate how random people's performance is. So firstly, for unique diagrams, we did not find a significant main effect of condition. But as you can see, people's performance with the active TMS to the different sites tended to be poorer compared to sham. For unique trigrams, we found a significant main effect of condition, where people's performance at multiple sites, at several of the sites, was significantly poorer compared to sham. So based on these performance outcomes, we then asked ourselves, how can we use these to identify an individualized site based on people's performance? And so we looked at using this ranking methodology shown here, which is quite simple. It's just comparing people's performance at each of the active sites compared to sham, and then using these performance outcomes for both main outcome measures to identify individualized site. And this was successful in determining an individualized site in the majority of the study participants. We then tested to see how this site performed compared to a standardized sites, that's F3, and the 2 other control conditions. And as you can see, and what you'd expect, is that people's performance was significantly poorer with both with the individualized site compared to these control conditions for both outcomes. So getting back to the original question, can we use a cognitive task to potentially individualize the site of stimulation at the left dorsolateral prefrontal cortex for using TMS to enhance cognition? So firstly, consistent with the earlier studies in the 90s, we found that if you apply active high frequency TMS during task performance on this random letter generation task to the DLPFC, that it actually reduced people's performance and people perform less random. Secondly, what we found is those inter-individual differences in terms of the magnitude of this effect across the different sites. And then we use these performance outcomes to develop our ranking methodology, which was successful in identifying a single optimized site in the majority of the participants, where when we tested it at the group level, this was associated with poorer performance for both outcomes. So the question is, what do we have to do next to further develop this methodology? Well, we're currently doing a prospective study where we're aiming to replicate these findings and also prospectively test to see whether using an individualized site using this methodology produces greater cognitive enhancing effects in healthy participants than what we'd typically expect. Thank you.
Thank you, Donel. I think this is a very interesting concept with high clinical implications. So we have a question from Nigel. So have you tested whether the optimal site within an individual is stable over time?
So that's a very good question. I guess the same thing applies to with functional neuroimaging targets. So no, we haven't done a test-retest reliability analysis, but what we're aiming to see is whether we get similar results in a separate sample of people.
Yeah. Excellent. I have a similar kind of question. So could you tell us about the future direction in terms of the the next step, like combining with MRI, fMRI, like as Robin did?
Yeah, exactly. So that's the next step. So we're planning on looking at either looking at the network effects with fMRI when we stimulate this site relative to either a standardized site or a structurally based site, looking at neurophysiological outcomes, so looking at kind of like EEG measures and looking at network changes with this site compared to other sites, in addition to looking at conducting prospective studies where we examine the efficacy of this site for producing cognitive enhancing effects. So yeah, so there's a few different directions we can go in.
Great, thank you. Now, Donel, one last question from Kate. So how many of the participants showed the expected pattern of response?
So in this sample, we saw a majority of participants showed decreased performance, but of course there is some variability, and as with any brain stimulation experiment, and some people showed the opposite effect. But at a group level, it was as hypothesized.
Excellent, thank you. Now, I think it's time to wrap up this session. So I would like to say thank you to all the presenters for the excellent presentation, and also thanks to the attendees for your attention and contribution.